Hello there, welcome to another lesson on Beth series Biologists Explain This. Good morning, good afternoon, good evening, depending on the part of the world you are listening to me from. I still remain your biology tutor, Peter Sabiodun, and in today's lesson, we'll be looking at cofactors. Now, we see so many things often about enzymes as though they worked alone. So, in this lesson, we'll be shedding more light on molecules that help in catalytic activity of enzymes, such that without these molecules that we call cofactors, catalytic action of the enzyme will have been impossible or inefficient at worst. So let's look at the issue of cofactors. That's what we have been looking at in details in the course of this study. And I'm sure at the end of this study, you will have been very enlightened and informed that you want to go over the lesson over and over again. So come along with me. Let's enjoy this study together. Now let's first look at our assessment objectives like we always do. At the end of this lesson, you should be able to define what cofactors are, describe the different categories of cofactors, and be able to describe the roles of cofactors in enzyme activity, and also be able to describe the role of vitamins in enzyme activity and in the maintenance of good health, very important. And at the end of it all, be able to apply the concept and the principles you have learned in requisite exam situations to your success. So let's look at types of cofactors from this chart. Cofactors can be ordinary ions, that's metal ions, or they can be coenzymes and prosthetic groups. Now, the introduction here is that you can have ions and organic molecules acting as cofactors. Now, to your right, okay, on the screen, coenzymes and prosthetic groups are both organic in nature, and you've got the ions to your left as ions, meaning as inorganic. So as we proceed with the study, we are going to be seeing the details about all of these, each of these cofactors. But from this outset, you can see that coenzymes are loosely bound, meaning it's only in the course of the reaction that you see the coenzyme being fitted into the protein structure of the enzyme molecule, while the prosthetic group is always permanently attached to the protein structure of the enzyme protein. And still on the types of cofactors, we see this division again, this subdivision, showing you that you've got the metal cofactors, which are inorganic in nature, and you've got the organic ones, which are the coenzymes and the prosthetic. Just shedding more light on the subdivision so that you can quickly grasp in your mind's eye and you can quickly summarize them. So when preparing for the exam, in preparing for an exam, charts are always good because they simplify and they connect concepts in a topic. So let's look at introduction here for our purpose. Now, coenzymes are a type of cofactors. So that's to clarify a misconception here. All molecules that aid or assist or help enzymes in their catalytic activities, you call them cofactors. So from the two charts you saw in the previous slides, you saw that, okay, that coenzymes are a type of cofactors settled. And then we said also in the previous slide when I was giving narration that coenzymes are loosely bound to the enzyme, unlike prosthetic groups that are permanently attached. But there is something else that we want to also shed more light here. Now, the enzyme without a cofactor is ineffective. In that form, you call the enzyme an apoenzyme. Okay, so it's just like a hunter without its gun, for example, or a farmer without his cutlass, okay, or a sickle with which to harvest. And so, if you're going to hunt, with what are you going to hunt? An enzyme is ineffective in the form of an apoenzyme, and so without its cofactor, you call an enzyme an apoenzyme. And once the cofactor becomes attached to the protein structure of the enzyme molecule, it becomes activated, and so we call it in this active form holoenzyme. So in summary, holoenzyme is the active form of the enzyme because of the cofactor cofactor attached to its protein structure, while the apoenzyme is the enzyme in its inactive form because it's not yet activated by the attachment of its cofactor. So let's look at cofactors in closer and more details now. Let's look at them closely. Now, your cofactor can be organic or inorganic, okay? And then organic cofactors can be divided into coenzymes and prosthetic group. Still on cofactors. Now, there's something we want to explain here. Now, many organic cofactors are vitamins or derived from vitamins. I'm sure in your studies on aerobic pathway, when you are discussing electron transport chain inside the mitochondrion, you came across these molecules: NAD, nicotinamide adenine dinucleotide, and flavin adenine dinucleotide. These are cofactors. So in case you didn't come across them in your aerobic discussion, this is what you are coming across here now. So in answering questions to describe the role of these two molecules in the body, you have to first of all introduce them as cofactors, okay? And they are derived for vitamins. So NAD is derived from niacin or nicotinamide, and then you've got the FAD derived from riboflavin respectively. So they are derivatives of vitamins. And then a lot of inorganic cofactors are minerals of metal ions, and then they can be in their oxidized or reduced form or state when they are playing their role in assisting in the catalytic action of the enzyme. Now let's have a closer look at coenzymes as a kind of cofactor. Now we have already introduced the concept of cofactors. I want to just look at them individually and separately in much more details. Now, coenzymes are small, non-protein, organic, okay? They're organic or metallo-organic molecules, and they mediate in carrying atoms or groups, okay, in the active site. And so their role is to help in transition, okay? They help to transport reacting units between the substrates that you have in the active site. So they help in conveying this. And remember, in our previous lesson, we discussed catalysis, and we said the R groups in the active site, they help to cleave. We need to weaken the bonds that hold the substrate molecules together. And so the R groups are involved here. So the R groups can't do it alone. When the coenzymes get involved, they help in transporting these individual units, these atoms or groups between the R groups and then the substrate molecules. And then the vitamin Bs are very popular coenzymes that either they themselves or their derivatives, and then they are required to form a lot of molecules, fats, carbohydrates, and proteins. And then you've got this adenosyl methionine, which is very important in transferring methyl groups, okay? And it's commonly used by virtually all organisms. So prokaryotes like archaebacteria, even generally bacteria. So the eubacteria, archaebacteria use methyl groups, and then eukaryotes, also like humans, also use this. So it's very common to understand that there are coenzymes that can also cut across all organisms, apart from the ones that are peculiar to different organisms, as we are going to see as we proceed with this study. Still on coenzymes. Now, we said, like, coming from the previous slide, that they donate, okay? They can either be donors or acceptors of electrons from inorganic molecules or chemical groups during the catalytic action of the enzyme. Still talking about what you have going on in the active site. The active units in the active site are the R groups and the substrate molecules. And so these are the ones that are involved in the catalysis. And so the role of the coenzyme is to help in transporting the chemical units, okay? They donate or they accept. So if you look at NAD, for example, NAD in its oxidized state, okay, it's willing to accept hydrogen, okay, and electrons. And so when it gets involved in reactions, NAD oxidized, but that with a plus becomes reduced, NADH, when it has accepted, and along the pathway, you see NAD transferring hydrogen to another molecule. So it's acting as an intermediary, okay, accepting hydrogen and transferring hydrogen. That is the role of NAD. So it's an example of what you have as the role of coenzymes. And then to your surprise, you may realize that ATP, very popular. I mean, ATP is popular when you discuss energy concepts in cells, whether in microorganisms or eukaryotes, or you are seeing here that even ATP is a coenzyme. Fantastic information there, very, very, very enlightening, that ATP and ADP derive from it also because they are interconvertible. If you hydrolyze ATP, you are going to get ADP and inorganic phosphate, and if you condense ADP with inorganic phosphate, you're going to get ATP. So these two are coenzymes, and they are very, very involved in energy processes. So in the process of energy pathway, you're going to see them a lot. And then you have some very important molecules called iron sulfur clusters, okay, the metalloproteins, and even DNA. Deoxyribose sugar, okay, in DNA is also a coenzyme. So you are going to see, so what you are seeing in the picture is that if we join this topic to what you have learned so far under aerobic respiration, or let's say energetics generally, energetics talk about both photosynthesis and respiration in cells. So you are seeing all the molecules you have been hearing about. You are seeing their molecular nature and why they are able to play the roles they play in the topics you have learned, and you are still going to learn about them. And then we can classify coenzymes into co-substrates, and of course we have the contemporary that you have, prosthetic group. Now, it's not really a classification, but it's a description here now. Now, if you look at the organic cofactors, you've got co-substrates which are temporarily attached in the course of the reaction only, okay? And then examples of that, you've got your NAD, okay? So the molecules that you have playing the role of co-substrate and prosthetic group, they are part of the discussion we are having. Now, a very interesting prosthetic group is heme, okay? Heme is a prosthetic group that is attached to hemoglobin molecule. Heme helps to bind to oxygen in hemoglobin, so that's its role, and so it helps to transfer oxygen between the reacting units in the aerobic pathway. And of course, I've already spoken about NAD. NAD is the hydrogen carrier in aerobic pathway, while NADP is hydrogen carrier in photosynthetic pathway. So if you have not yet studied aerobic respiration or photosynthesis or energetics as an energy related topic, you're going to learn this later. Both NAD and NADP, they transfer hydrogen. So in their reduced state, you see them as NADH and NADPH respectively, and that also includes FAD, which becomes FADH when it becomes reduced. Now let's talk about vitamins, because they are very important cofactors. Now, vitamins are very important organic substances that act as cofactors to help and assist enzymes in their catalytic activities. And then the body naturally is unable to produce vitamins. So if you've learned your previous under nutrition, okay, under heterotrophic nutrition, you must have come across essential and non-essential vitamins, just like you learned about essential and non-essential amino acids. So the body generally is unable to produce vitamins, and so they always have to be available in our diet. So our diet should have enough of vitamins. Now, if you've heard about vitamin A, for example, being needed for good vision, vitamin A is not actually present in the body. It's not available as vitamin like well. So for you to have vision, vitamin A has to be metabolized. So when you eat food that contains vitamin, okay, it's metabolized into retinol that helps our vision on the retina. So it's important to realize that all these vitamins are important. Now, see the importance of vitamin C, for example, in the synthesis of collagen, because vitamin C generally are involved in biochemical reactions. They get involved in helping. Vitamin K, you're going to see vitamin K when you get to blood clotting. Vitamin K is important in the pathway of blood clotting. It just gets involved to ensure that the blood clots, so that the person injured does not overbleed, and then the site of infection is not prone to infection by pathogens. And so it's important to realize all of these concepts about vitamins, and we are still going to say more about them in subsequent slides. And we are looking at a very important vitamin called thiamine, also known as vitamin B1. Now, it's important to realize that it's converted to thiamine pyrophosphate, which is required in the metabolism, that is, the pathway in which carbohydrates and amino acids are being used by the body. And it's present, of course, it's common. So if you have a deficiency, anybody with vitamin B1 deficiency, it's rare because it's abundant in rice and corn and beans, and these are staple foods of different parts of the world. But it can be added also to enhance the vitamin value of the food, in particular cereals, your cornflakes, your rice crisps, and the rest of them. The companies add artificial thiamine to help the health of the child particularly. But there's something else that I just want to touch briefly, and that is the issue of the Korsakoff syndrome. Okay, now this syndrome is connected with the alcoholism. Okay, and then it's common to see individuals who are deficient in thiamine having this also. Now it's alcohol intake, the addiction of alcohol that leads to this syndrome. Okay, it's an encephalopathy. Now, when you talk about pathy, for example, pathy as pathy as a suffix, when added to anything, means suffering. Okay, so something like sympathy means we suffer together. Okay, and then so you've got neuropathy. Okay, you've got myopathy, and the rest of them. You've got retinopathy, because when you add pathy to anything, you're talking about suffering or a disease in that part of the body. And so this particular syndrome affects the psyche of the affected person. So it's a neurological condition. So there's anxiety, there's fear, there's change in identity of the people. So you can see that the vitamins are not just what you take, they are very important in the health of you and I. Our health is very important. So you can read more about this Korsakoff syndrome, because it talks a lot about the fact that the mental state of a person can be affected if you don't have enough vitamin B1. Having talked about vitamin cofactors, let's talk about non-vitamin cofactors. I made mention earlier on about ATP, and ATP, what we know as adenosine triphosphate, is organic, but it's not vitamin. It's a cofactor. You often see it providing energy in various pathways. So in energy pathways, everywhere energy is required, ATP is regarded as the currency with which cells trade. Now let me clarify this here. The food you and I eat does not supply energy directly. Let me say that again. When you eat your food, when your food is eaten, is first digested. Okay, so your starch in form of yam or bread or pastry, whatever you might have eaten, is first digested from the polymer to the monomer, meaning the simplest form, and then the sugars resulting from the hydrolysis of starch will now be fed into the energy pathway of aerobic respiration. So it is from there that your glucose is completely broken down to CO2 and water. So energy does not come from food directly. Food is first digested in the alimentary canal, and then the soluble end product, so the amino acids from protein digestion, fatty acids from lipid metabolism, and glucose or sugars from carbohydrate metabolism, they are the ones used in energy release. So it's very important to understand that the ATP is not just to give energy. It's important because it transports a lot of units. It's within the reaction site of the enzyme protein, and then you have got coenzyme Q. If you study electron transport chain and the mitochondrion to end aerobic pathway, you must have come across cytochromes. Okay, now there are so many coenzymes there in the pathway, and one of them crucially is this coenzyme Q. It's very important in the pathway of electron transport chain in the mitochondrion. And then I spoke earlier on about him, a prosthetic group, okay, that you have in hemoglobin protein, helping to bind to oxygen. So these are examples of non-vitamin cofactors. Now let's talk about a very important um, cofactor called folic acid or folate. That's vitamin B9. So you can begin to picture the vitamin B's, okay, that you have. Very important. There are so many vitamin B's, a lot of them. I'm just picking some of them for the purpose of this lesson. Now folate, or what we call folic acid, okay, it's very important in the process called erythropoiesis. Now erythropoiesis is the process whereby new red blood cells are being produced. Now it will surprise you that the part of the body that secretes erythropoiesis is the kidneys. So the kidneys are the organs. So you'll be wondering, so when you hear about glands behind glands, so the kidneys can play a role of glands. Yes, there are many glands in the body. Even in your stomach lining, you've got cells that secrete chemicals also. So the erythropoiesis, so the chemical secreted from the kidney is called erythropoietin, and erythropoietin stimulates the bone marrow tissues, bone tissues, to begin to carry out red blood cell production by the process called erythropoiesis. And so, and very, very important, when a child is developing, the brain, the skull, the spinal cord has to develop properly while the child is in the womb. And so a pregnant mother is supposed to take a lot of folic acid for the proper brain development of that child. A child whose mother did not take enough folic acid may be mentally deficient after birth. And also we are talking about the fact that the DNA and RNA, our genetic molecules also, they are produced from folic acid. Folic acid is involved. So it's very important to realize that you and I need a dosage of folic acid daily or regularly to enhance our brain development and to ensure that our red blood cells are at a particular optimum level for maximum oxygen transport in the body. Now let's talk about inorganic cofactors here. So we've seen the ones that are vitamin based, the ones that are non-vitamin. Now we are looking at the inorganic cofactors. There are a variety of them: magnesium, you've got manganese, you've got zinc, molybdenum, and copper. The important thing is that these can also act as cofactors, helping. And in particular, one example you see there is the fact that coenzymes or inorganic coenzymes, or sorry, inorganic cofactors can bind to the allosteric site of the enzyme. They bind allosterically, and when they do that, of course, like we've seen, the shape of the active site is distorted. Like we said, this can enhance or help the induced fit mechanism of enzyme activity, where you bind and then both the enzyme's active site and their protein substrate molecules are adjusted for effective binding, so that the AES complex can be formed and products can be formed through that binding. And then, still on inorganic cofactors, we've got the issue of the fact that these inorganic cofactors, they can help to expose the R groups of the amino acids, because if you are going to have catalysis to weaken the bonds of the substrate, you've got to have all these R groups rightly positioned in a particular way. So when the substrate is coming into the active site, the substrate molecules are orientated in ways that their positioning exposes their bonds to the R groups. This can happen by allosteric binding of these inorganic cofactors. They can alter the shape of the active site to expose the R groups, so that the R groups can properly interact with the approaching substrate molecules. Still on the inorganic cofactors, we are seeing the fact that the prosthetic groups that we have, they are family bound, of course. So we're rounding off now very soon. Now these inorganic, of course, we are treating inorganic, so just look at what I'm discussing. Now the prosthetic groups that you have as cofactors, they are bound permanently to the APO enzyme, and they cannot be removed except you have to apply heating or any other means. So since they are permanently bound to the protein structure of the molecule, unlike coenzymes, prosthetic groups as cofactors, they cannot be removed until or unless you want to damage that protein molecule by denaturation, which is not needful anyway. And so here we are discussing the fact that prosthetic groups contain the ion metal. Okay, so the important thing about these ones is still emphasizing the role of ions, and so ions can aid prosthetic groups. Now Prosthetic groups like heme, for example, contain metallic ions. Now, hemocyanin, which is found, which contains copper ion, is found commonly in crustaceans like your crabs, your crayfish. Now, these organisms, their blood is blue, unlike ours that is red. I'm sure you've learned from your discussion on the circulatory system that blood doesn't have to be red. Let me say that again. Blood doesn't have to be red. Our blood is red because of hemoglobin protein in our blood. So what makes blood to have a particular color is the ion that's in it. So hemocyanin, okay, the copper in hemocyanin gives this pigment blue color, while iron in our blood, in our hemoglobin, gives our own blood red color. And there's a particular molecule that you find in plants. Okay, since we are discussing cofactors that contain ions, okay, plastocyanin is a copper containing pigment in plants, and it's very important in the process of photosynthesis. So just look at these two diagrams, very vivid and very clear and very informative and enlightening, trying to talk about what the way cofactors aid enzyme activity. Look at the shape of the apple enzyme and then see what happened when the coenzyme fitted into it. It just made it possible for the substrate to form a perfect fit. So but the two diagrams you're seeing, they elucidate, they shed more light on the fact that cofactors are very crucial. Now, this particular cofactor is a coenzyme. It's binding temporarily, so it's binding to that apple enzyme just in the course of the reaction. And so, as we round off this lesson, like we always did, let's assess ourselves on the questions you see on the screen. Go through them, answer them. You can always go over the different slides over and over again. And then, like I always say, be free to call on me, to reach me on any of these questions. So practice the questions at the end of our lessons, or as you are preparing for your examinations, you come across questions, please, you can get in touch with me through my WhatsApp, my email, my Instagram, and on YouTube. We can discuss together. And if you're interested in online tutoring, I can partner with you on the journey to your success in that examination. Remember, we work together. We at BET, our focus is simple, is to help you to achieve success in that examination. We hate and we propose to remove fear and anxiety and build your confidence towards that examination. If you have enjoyed this lesson, please subscribe to our YouTube channel. Okay, subscribe to our YouTube channel, like and share, and then share this with your friends. Like, tag your friends, share it, and in particular, I want to hear from you. I would like your comments. Comment observations are very welcome. If there are topics that you want us to take, let us see them in the comment section. If there are things that you want us to adjust, just tell us. We want to hear from you. If you are enjoying us, we can do much better than we have been doing up till now. And so if you have enjoyed this lesson, I will hope to see you in the next lesson, and I hope to hear from you. And again, please like and share and comment and send in your responses to our questions and subscribe to our channel. See you next lesson. Bye.